almost afternoon, everyone, ladies and gentlemen. And thank you very much also for Professor Matsuda Sensei for the talk. So we learned a lot from your talk. So probably in this time, so I would like to share a little bit about our work in the development of Indonesian local COVID-19 vaccine in IDB. So this work actually is multi-faculty works. Yeah, so this is works from Ibu Alucia Anita and Tia and Ibu Devi from School of Pharmacy. Also Ibu Desi, Ibu Iis, Ibu Ferni, Ibu Fifi, and Yofin from Chemistry Department are also involved. Ibu Marcelina Irasoniatan and Intan from School of Life Science and Technology. So this is a very multi-faculty research actually in our, and we also belong to Research Center of Bioscience and Biotechnology, ITB. So as we know so far about the case about COVID-19, it's already given a big impact actually to our lives. And it's already infected around 240 million people all over the world. And we can see in here that a lot of effort has been put on it, also with the vaccination. And until now, it's already a 47.7% of the world population has received at least one dose of COVID-19 vaccines. But still, this is the vaccines, we need more and more to fulfill the demand of the vaccination itself. And we can see in here also only 2.5% in low-income countries have received at least one dose. So there's a gap actually between the developing and developed countries. And we can see in here that the need of vaccine is still a lot. And how about Indonesia itself? So in Indonesia itself, actually it's already infected around 4 million people. And we can see in here about the vaccination itself, the government has already put a lot of effort on it. And the vaccine itself come from the imported vaccines so far, and it's already 101 million people received the vaccine at least in one dose. So we can see in here still a lot of doses of vaccines needed in Indonesia. It's around 208 million people need to be vaccinated, and a lot more dose needs for this vaccination. Yeah, so to overcome this problem actually, so there's a lot of effort from the government. So there's a lot of companies has been cooperated with the government itself to fulfill the needs of vaccines. But there's also effort from the local, how to support about the needs. But if we would like to see the big picture of the vaccine itself, the need of the vaccine itself, like Professor Matsuda Sensei has already give to us, there are several platforms for the vaccine itself. So there is protein-based vaccines. So there's a viral vector vaccines, there's nucleic acid vaccines, and there's a virus vaccines. So for the virus itself, so there's an inactivated and also there's an attenuated. But in here, so we can see there's some technology which is become a latest and recent technologies like a viral vector and nucleic acids. For protein-based itself, some of the vaccines has already been generated with this platform. So this is a few platform for the vaccine itself. And as we know, after it's injected, so this vaccine in our body, so we'll meet with antigen presenting cells, and these antigen presenting cells will degrade the vaccines and then present into the surface of the cells, and then it will become recognized with the T cells, and afterwards for the downstream effect, it will produce the antibody as a humoral immune response or cellular immune response. And we can see in here that the COVID-19 vaccine itself has already been produced by several companies. and is already delivered and received by few peoples in the whole of the countries. And we can see in here, there's vaccines with RNA and DNA vaccines, has already been approved, and adenovirus also, and inactivated, and also subunit vaccines. And if we can see in here about the development of vaccine itself until now, if we can see the data from the WHO, the development of the vaccine itself is very, very growing, so vast. If we can see from the first case of COVID-19 from the December 2019 until now, so people try to put an effort on it, and then we can see in here there is a lot of development in vaccines. For example, in the clinical development is already 126, and the preclinical development is already 190 for vaccines to be developed. And we can see in here there's several platforms that has been developed so far. And we can see in here the number of vaccines, for example, for clinical development, we can see there's a few different platforms as we discussed before. So there's a protein subunit here. So the protein subunit is becoming the first platform that people interested to develop. So this one, the protein subunit, and there is a viral vector itself. In here, the non-replicating, in here, and there's another platform itself. So we can see in here, there's several different platforms, and there's already a lot of platforms developed by all of the countries in the world. So, so how about ITB? So ITB itself actually is part of the consortium vaccines called Vaksin Merah Putih. So in our institution, we try to develop actually the vaccines with two different platforms. The first platforms that we would like to develop is protein subunit vaccines. And the second one is the adenovirus. So why actually we try to develop this platform, so we can see later. So why we interested to develop a vaccine candidate based on protein subunit? This is because for the previous experience, we've already have such an experience about how to develop the vaccine candidate in a protein subunit in the lab scale. So start from 2000, the development of candidate vaccines start from a group of Ibu Devi Retnoningrum. And then afterwards, so we try to develop in the consortium vaccines with the hepatitis B vaccine consortium, start from 2012 until 2017. And we've got several prototypes in that time in the laboratory scale that's already proven to induce humoral and cellular immune response. This is a collaborative research actually with several different institutions. So lead by Biopharma, and then involve several institutions like Eijkman and also BPPT. So in that time, we have a different prototype like a seed from Pichia pastoris and also E. coli that can produce a hepatitis B surface antigen and hepatitis B core antigens. And in that time, we also has produced one of the expression system from Hansenella polymorpha. So, and then we also some experience to work with collaborative research with US to develop intranasal administration of chimeric HPV and norovirus vaccines. So based on this experience, we try to develop actually the COVID-19 vaccines. So in the protein subunit itself, so this is the vaccines with the protein subunit, which is developed with the lead of Ibu Desi Natalia, Professor Desi Natalia from Chemistry Department, and his team in here. So what Ibu Desi Natalia and team try to develop is try to produce the RBD, the receptor binding domain, which becomes a part of protein spike, like we heard from Professor Masura Sensei before. There's a spike protein which interact with the receptor ACE2, and receptor binding domain is becoming a part of protein spike, and then try to develop by this group through express this protein in E. coli and yeast. So if we can see in here, for the construction and production of RBD receptor binding domain in E. coli. So what they are doing is they constructed the recombinant RBD in the RBD plasmids, and then they put into the E. coli, then overproduced, and then they purify and test the immunogenicity study in mice. So what they got is in here, so they've got the RBD protein, which is pure RBD, and they got it from the inclusion bodies first. And afterwards, they make a refolding, and then they got the pure protein in here. So we can see in here is quite clean. So they have successfully purified this protein, and then they inject it into mice. So this is the work done by Ibu Marcelina Tan in here and the team. And we can see in here from the result that this receptor binding domain is stimulates the antibody production. So we can see in here, there are 3 times of injection. So this is a prime injection, and then 2 boosting in here. And for several days, the blood is taken and then is sacrificed in days 63. And we can see in here, for the group mice, comparing with the control, male or female or male here. So we can see that the titer antibody with the injection with the RBD is higher. So we can see in here the result in male and the result in female in here. So we can see that for both of the groups of mice, that the RBD that is purified before and constructed before is already being produced. And then it can stimulate the immunogenicity. And we can see in here that the immunogenicity is good for a group of mice. So that's so from here, so we can see in here that for the conclusion for the first one, so we can see in here that the RBD derived from E. coli has been generated and immunogenic in mice. So besides that, so in addition to the work with the RBD in E. coli, Ibu Desi and her group are also try to express the RBD not as monomer. From here, the RBD is a monomer, but we can see in here, even though it's in a monomer, it already can induce the immune response. So it can induce antibody response. But afterwards, they try to express the RBD with the fold-on. So as we heard from the Masura Sensei before, that the protein spike can form the trimer. So that's they would like to mimic actually the RBD to become a trimer by using the fold-on. So this protein fold-on, actually the protein that resides from the fibritin T4, and this is the fold-on that could be used for make the protein becoming trimer. So this is done by Kenny, with the help of Kenny. So this protein is, the gene for express the protein is constructed, and then it's trying to be expressed. So for the first time, before they're doing in the yeast, they are trying to express actually the trimer RBD with a fold-on in the E. coli. So we can see in here, So they are successfully actually to express this protein, the RBD with the fold-on in E. coli. So we can see in here, so this is the monomer, around 26 kilodalton, and the trimer around 78 kilodalton. So with this work, so they convinced actually that this protein could be expressed well. And afterwards, so they try to subclone actually to the plasmid that can be used for expressed in yeast, Pichia pastoris, and Hansenella polymorpha. And the results so far shown that this protein also could be expressed in yeast, in Pichia pastoris, and is still being constructed in Hansenella polymorpha. And we can see in here that RBD fold-on has been expressed in E. coli, and for Hansenella polymorpha is still being constructed. However, for Pichia pastoris is already being expressed but still being optimized. So this is the result for the first platform, for the subunit platform for E. coli and yeast. And the second one, actually, we are also trying to generate the other vaccine candidate platform based on adenoviral vector. And so for this one, the work is done mostly by Ibu Anita Aulicia Artarini with her team, Tia. So Ibu Anita is a prominent scientist, and so it's good, doing a very nice job in here. So we can see in here, the work itself is starting from the beginning actually. So we're trying to construct the plasmids with the codon optimization. So it's the same that Ibu Desi and group as well. So they are trying to codon optimize actually the construct in here. And after they're sure about the construct itself and they check already, so they try to make the recombination in here. So they try to make the recombination with adenovirus genome. So the plasmids, which comprises the protein spike in here, so they make the plasmid, the protein spike with the codon optimization, the protein spike derived from SARS-CoV-2. And then they've got the plasmid itself, it's already checked and it's already sequencing. And afterwards, they try to recombinant with the genome itself in here. And afterwards, after they got the recombination of the adenovirus with the protein spike, they transfect into the cells, the HEC293. So they would like to produce actually to transfect to get, to make the virus, to make the adenovirus, which has protein spike on it. So in here, so we can see, so this is a very nice result actually, because we can see in here, this is the cells that can infect it. So the brown in here is the cells which is infected with the adenovirus. So the adenovirus is signed by the antibody against protein hexon that could recognize hexon from the adenovirus. So we can see in here that the brown here shown and revealed that the transfection of the adenovirus already successful. And we can see in here, this is the primer, the PCR result for the gene of protein spike. So we can see the positive result in here. So we can see that from this experiment, we can see the result of the adenovirus, which has the protein spike on it. The gene that encoded the protein spike on it. And afterwards, there's doing the amplification, virus amplification, and And also the virus is amplified for immunogenicity study and then purified and studied the immunogenicity study in mice. And after the purification itself, the adenovirus, which has a protein spike on it, so it's studied the and then going to the immunogenicity study. So we can see in here, yeah, so this is from the previous result. There is an affected cells with the adenovirus on it. Yeah, and we can see in here also that the protein spike can express, this is the expression of protein spike. So the adenovirus, yeah, which has the protein spike, can express the protein in here. So this is the cells which is infected with the adenovirus with a gene that encoding the protein spike, and this is the result of the protein spike itself. So we can see in here we have the adenovirus, and the adenovirus can generate the protein spike, yeah, which can induce the immune response actually for the COVID-19. And so we can see in here from this experiment that the adenovirus itself could be amplified in HEC293 cells. However, so we would like to generate the adenovirus which not replication competent in our cells. So this the model to test that the replication incompetent, yeah, is done with the A549 cells. We can see in here, yeah, so actually what we would like to have is the adenovirus that can be produced as an adenovirus with protein spike, but if injected into our body, is becoming incompetent for replication. So it could then replicate after injected. So this is the model of the experiment that we're doing so far. We can see in here the cells is still alive, yeah, but there is no replication of adenoviral in here. So but if we can see in here, there is an expression of the protein spike. So this result is very encouraging because we can have adenovirus with the protein spike expressed in here. Yeah, but this adenovirus is also could be produced to become a vaccines. But after injection, the adenovirus becoming replication incompetent. So it would not be a replicate after the injection, but still can express the spike itself. And afterwards, we are trying to produce do the immunogenicity study, and we can see in here for the preliminary results done by Ibu Marcelina also with her team, that adenospike stimulates SARS-CoV-2 antibody production. So this result is still being completed, but we can see the encouraging result in here. We can see this is adenovirus, which can express the protein spike. Yeah, we can see the differences. So this is the control, yeah, without adenovirus, and this is the control with adenovirus empty, without the protein spike. And in here, we can see after the vaccination, 14 days after the vaccination, we can see the titer of antibody is quite high in here. So from this result, so we can see that we can get the adenovirus that could express spike SARS-CoV-2 that has been generated and could be produced in HEC293. So this is encouraging result for the production for spike SARS-CoV-2, adenoviral with a spike SARS-CoV-2. And also this spike replicate is replication incompetent. So meaning after the injection is quite safe because it couldn't replicate after the injection. And the preliminary result, we feel that adenospike is immunogenic in mice. So what next afterwards? Yeah, so after all of those, yes.
Dr.
Okay,
Erna,
okay.
time is almost up.
Okay.
Only 30 seconds.
Okay. So this one is just the next to do. So we have to do the scaling up and also the characterization of the profile product, and we can move to another session. And then this is hopefully all of the components that we've got in here, so can become something to overcome and to face the next pandemic. And this is for the funding. Thank you very much. Thank you, Dr. Marcelina.